Hi everyone, my name is Luhan. I'm the postdoc from Church Lab, also the co-founder together with George for the startup eGenesis. Today I'm happy to present to you our progress of using mammalian genome engineering to deliver xenotransplantation as a safe and life-saving procedure. So here we are looking at the number of the patients suffer from the end-stage organ diseases in US. So there are over 2 million patients suffer from the organ failure. Only about 123,000 of them are putting on the waiting list. And only 1/10 of them can get transplantation each year. So there's a huge delta and human life is at stake. The situation is worse in other parts of the world, especially in Asia, when there is no donation culture. So hypothetically, there are probably 3 ways to solve this problem. One, we have more donation. Second, probably as Alex introduced, we can use stem cell to produce an organ. And the approach we take, which we hope can help us get there faster and quicker, is to utilize the animal as a bioreactor and change in the way that human compatible. So the process works in the way that we start by engineering the somatic cell. Then as people clone Dolly, we clone transgenic pig from the engineered cell, then deliver organ to the patient. We didn't invent this idea. So people have pursuing this for more than 2 decades, and big pharmas invested very heavily in the 1990s, but it's a huge failure. And there are 2 reasons why it didn't work. First, as you can imagine, there are huge rejection issues between the pig organ and human body, and people underestimate how many antigens they have to remove, and there was no tool to do that. Second, people identify the endogenous retrovirus resigned in the pig genome, and if you culture pig cell together with a human cell, the virus will jump from the pig cell to the human genome. So it didn't work, potentially can become a plague. And that's where we think CRISPR and other advancement in mammalian genome engineering can come to play. And our lab was one of the first labs to demonstrate we can use CRISPR to engineer mammalian genome. We think the beauty of CRISPR is the multiplexability. With such a tool, we can conduct high-throughput engineering to remove the antigen, introduce the immune inhibitory factor to confirm the pig organ human immune compatibility. In addition, we can use the tool to eradicate the virus from the porcine genome to ultimately address the safety concern. So this is a study we published last year. We first utilized a sequencing technique to identify there are 62 copies of the endogenous retrovirus, in particular in the genome of particular cell line we are working at, and we designed CRISPR specifically to tackle the catalytic domain of the reverse transcriptase of the virus genome. And after the treatment, we got the cell line with 100% knocking out of those 62 copies of the virus. And here we are showing that if we culture the pig cell together with a human cell, without modification, there are still prevalent infectivity, whereas if we do that with our engineered cell line, there were minimum infectivity compared to similar to the negative control. So our work increased the multiplexability of gene editing in mammalian cell by almost 2 orders of magnitude. And more importantly, we demonstrate we can solve the biggest safety issue of xenotransplantation. After that, we move to our second challenge. How can we engineer the pig to have the immunology feature? And over the last 1 decade, there are some scientific progress that people produce different transgenic pig, and each one of them carries some modification. The good news is all the pigs are viable, and they all shows beneficial effect for xenotransplantation. But it takes years for them to generate those pigs, and some of the modification couldn't be implemented due to the lack of the tool. So over the last few months, we have generated pig 2.0 cells with 12 modifications, which we think can address antibody binding, complement coagulation, as well as the T-cell toxicity issue. In the meanwhile, with the help of the Denmark Aarhus University, we built the somatic cell nuclear transfer system in which we can take the engineered cell, put into the pig egg, and get a pig blastocyst in 1 week, which we are going to do the embryo transfer to get the pig, as well as also a very quick feedback to let us know what's the developmental impact of our modification on pig. So we are in the process of generating our pig 2.0, and every cell, every tissue, every organ of our pig will have the identical genotype. So we think our pig can serve as a bioreactor to produce different organ, different tissue, such as for heart transplant, liver transplant, pancreatic transplant, as well as neuronal transplant. And I hope I convince you that the advancement of gene editing really reinvigorate the field of xenotransplantation. In the meanwhile, I think if we can solve the immunology issue, we can do things beyond that. We can engineer the liver to be cancer resistant, as Jeff mentioned, virus resistance, as George mentioned, or produce some non-essential amino acid, as Harris mentioned, and we don't need to worry about the ethics issue if we need to use that directly on human being. So to a certain extent, xenotransplantation opens a back door for us. We can really exercise the tool to push the envelope of gene editing in terms of the scale and complexity, which may have the impact beyond our practice. So last, I'd like to thank George for being a visionary and enable everything happen, and my team. Specifically, Mark Guile led all the synthetic biology as well as bioinformatics effort. Don't build the embryogenesis system, and working together with Alina to lead the effort of the PIC 2.0. And I'm happy to take any question. Scott.
So in the paper where you described the inactivation of 62 loci at the same time, these were all PERV loci. Do they include all the PERV families, the C and the D, and I think there's an A, and do they hit all of them? And then in the cell type that you used to achieve this, is that clonable, that cancer cell line, PK15? Because I'd be interested if it is, obviously.
Yeah. So Scott asked two questions. First, whether we hit all the PERV family, and second, whether our cell line is clonable. The first one is yes, there are A, B, C, and A-C family, and we hit all of them. And there are some pseudogene, and we designed the CRISPR in the way that we only target those active PERV, not touch other part to prevent some genomic instability. And the second question, no, the cell is not clonable, but we are repeating our process in the clonable cell line and hopefully can produce a transgenic pig with such a feature.
Hi, Liam Holt from NYU. So I was wondering about the immune rejection problem, and I think you were talking about changing the antigenic surface of the cell, but also cells present peptides from the interior of the cell. I was wondering if you could break that pathway so that you aren't getting arbitrary pig peptides displayed.
Yeah, so Lian's question is, even you can remove the antigen from the pig surface, the cell debrief will be picked up by the antigen presenting cell by the host, then trigger the indirect rejection. And to solve such an issue, we are not only knocking out the antigen, we are inserting some of the immune inhibitory factor, for example, CTLA-IG, which can suppress the T-cell reaction and hopefully can mitigate such a problem. Okay, thank you.
Hi, I'm a postdoc in Pam Silver's group at Harvard Medical. I don't know what his chair is here. So I'm the postdoc working on the human artificial chromosomes in the lab. No one else is working on it. We're relatively new to the game, but I'm going to catch you up in 5 minutes. So why are we interested in HECs? There are obviously a diverse amount of commercial and academic interests. Nowadays, it's not so easy to differentiate between the two anymore. But we're really interested in HECs because, as many of you have mentioned, it will really hack into chromosome biology across all organisms, so not just humans, but it will really push the boundary on many different things. And if HECs get too controversial, I might go into lizards and do like dragon artificial chromosomes, but this is the goal, which is to hack chromosome biology. And so what makes a good hack? And in terms of building a synthetic genome, what do we need to do with a hack before we can do that? And there's a way to score hacks for us, and I'm going to go through the criteria. So the first really important thing is segregation. Having a hack that's centromere competent, and this is the core of chromosome biology. If you can't segregate your hack, it doesn't work. And the next thing is compatibility. And this is what gives hacks a leverage above viral plasmids or random pieces of DNA, which is compatibility with the system. And this is a lesson we can learn from yeast and bacterial plasmids and vectors. They work with the system, they come from nature, they are expressed, they are recognized by cells, but in human biology, we haven't breached that barrier that much. So we keep trying to use viruses and things that will be attacked by human cells will be silenced and degraded. And so our objective of the hack is to make something compatible. The next thing is organization. So we don't want to just create a jumble of DNA that you throw in and you don't know what happens to it anymore. You want to know what exactly is in your hack and what it looks like while it's in the cell and if it gets passed on with integrity to the next few cells. It has to be replication competent. If it doesn't replicate, it's not useful. And expression is also super important. If you can't express of your hack, it's not useful again. And I'd say that in the last 2 decades, we've made significant progress on hacks in terms of meeting these criteria. And now for our lab, we really want to hit the last one, which is shuttle ability. And what this means is being able to shuttle it, like yeast vectors, into bacteria or yeast and manipulate them, construct them, and then put them into human cells. And this will accelerate development of hacks so quickly and genome synthesis as well, because if you can use the tools we have right now that work really efficiently, building megabases of DNA in yeast and then shuttling it into human cells, that will make it so much more multiplex and high throughput than we could ever achieve right now. So I'm just going to talk about the last 2 decades of hacks and what they look like. And I'm really glad that Vladimir has gone ahead of me because now you guys are up to speed on hacks. So one way is top-down, like he mentioned. So taking a naturally existing human chromosome, this is the group from Japan. And what they did was that they chopped this up. They removed all of the bits in between the centromere and the telomeres. So all you have are the core and the ends. And this is now about 5 megabases. It's been truncated, and it's in chicken cells. The other way is to go from bottom up, so de novo, taking a small piece of centromeric repeats, for example, and then amplifying that and throwing it into human cells, and hopefully getting something that exists, like persists as a hack. And so this is what Vladimir's group has done. And both of these hacks are now in CHO cells where you can load genes on via recombination, and then use microcell MMCT to transfer those chromosomes into your cell line of interest. So this process is still in development, as has been discussed. But we're trying to sort of go back to square one and see if we can make more well-defined hacks maybe, or make it a faster, more efficient process. So while Vladimir has sort of gone through his illustrious career of making hacks, I'm going to talk about our modest beginnings of making hacks. I've been about a postdoc for a year and a bit now, and this is what we've done. I'm going to run through it. We've redesigned the hack and centromere. We've found a way to assemble it, and we've also made a way to deliver really large hacks, so megabases in size. So I'm going to run through our design of the centromere. Centromeres are really repetitive, and this has been a big problem in terms of avoiding recombination or rearrangement, and we wanted to minimize this as much as possible. We went back to this consensus sequence back in 1987, and we removed as much of the repeats as possible, keeping only conserved motifs and inserting lag oocytes. So in this case, you can see that many of the nucleotides can be varied. So the longest stretch of any repetition is actually just that lag O repeat. And we've made it so that the lag O repeats will recruit a fusion protein of any type. SNPA is one option, but there are many different proteins in the human cell that can help to establish centromere function. And this is our hack design. So it's built on a yak because, again, we wanted to be able to shuttle between bacteria, yeast, and human cells, and we want to build it in yeast because yeast is really good at assembling really large and complex sequences. So again, it's been a yak, and as you can see, the yak can be linearized by cutting in the middle of a His gene. This means that there's the option of making your hack linear. We also have selectable markers like GFP and a drug resistance gene that is also an origin of replication, as well as the centromere, which, as explained, has a way to self-perpetuate. Insulators prevent reorganization of your chromosome, and also human telomeres. So we're in the process of putting in the telomeres, but right now we're already testing a design with the centromere in it. It's currently only 30 kb in size, which is pretty shocking for a lot of people, but there are studies that have shown that you can work with much less. So a centromere as big as 5 kb in human cells is capable of being a centromere. And the way we've done it through assembly has been quite complicated. It took us about a year to make this hack because of all the repeats and the large centromere and telomeres again. But the best way to do it, in our experience, is through tire assembly, which is to throw all your fragments into yeast with overlapping ends. And this works on the scale that's much better than any in vitro method that is out there right now. Lastly, I just want to talk about some work that we've done to show that we can deliver megabases of DNA from yeast into mammalian cells. And this is a really big barrier because you can't do this just through lipofectomy. You expose your DNA to too much damage in the process. But this means that you can now start creating hacks of megabases in yeast and pushing it into human cells. And you can get this at a rate of 1 in 300 cells will get the hack when exposed to the yeast. So a nice thing about it too is that if you have yeast that have your hack, you can deliver proteins too. And my collaborators managed to give human cells herpes through this method. So they assembled the herpes genome in yeast and also delivered it with proteins into human cells. And they've shown that the cells get herpes. So this is a super non-fun way to get herpes. So right now, I'm going to run through it a little. But the key ingredient here is arresting the cells in mitosis because this removes the nuclear membrane. And it makes it easier for your hack to get into the human cell and get accepted into the nucleus when it reassembles. And so again, the rates, if you compare 12 kb hack out to a 1 megabase hack, there's virtually no difference. They're all about 1 in 300, or like 1 in 1,000 cells will get your hack. And this is super amazing because, again, you can make so much with this. And so that concludes my talk. I just want to say that we're testing the hack now. And this work has mostly been funded by DARPA and has been a collaboration between my lab, Pam, and John Glass at JCVI. David's been working on the yeast to mammalian cell fusion technique, and Jeff has been really instructive in the design of the hack as well. And I have recently got a master's student, so I'm not the only one building the hack and testing in the lab. And yeah, these are my acknowledgements. Yep. Thank you. Question?
First of all, that was a marvelously clear presentation.
Thank you.
Including the slides, they were great.
Thanks.
I wanted to ask which different types of mammalian cells have you done this in?
Mainly we're testing in hack first, just because it's so easy to transfect. And we have protocols for the yeast to mammalian cell fusion optimized for hack cells, viral cells, even mosquito cells. So the protocol is really easy, and you can achieve rates of 1 in 1,000 delivery. But first, we're doing hack cells, but we'll definitely branch out into other ones once we troubleshoot the hack. So I don't expect it to be an overnight miracle. It's going to need some work. Yeah.
Normally, formation de novo kinetach or human artificial chromosome is accompanied by integration into chromosome.
Mm-hmm.
So only a fraction of cells contain HAC if you use highly competent alpha satellite DNA, and even loading of SNP protein, for example, loading factors. Did you observe integration of your cassette into chromosome?
So we haven't gotten that far yet. Up till today, we've just transformed it in, so it's been a week since then. I haven't been able to check whether they've integrated into chromosome yet. So right now, we're just trying to check if it can perpetuate for a couple of weeks first. But that's a really good concern because you don't want your hacks to just randomly insert into your genome. Yep.
Did you say that there's sorry, I'm Leslie Mitchell. Did you say there's a 5 kb centromere that's functional? Did I hear that correctly?
No. So we've just made our 5 kb centromere. You mean other studies? Yeah. So what people have done is that they've made about 5 to 10 kbs of lag-all repeats and thrown that into different human cells and tried recruiting different proteins through like a SNPA or HGP, other centromere projects. And they found that it can create a centromere, form a kinetochore.
So then there's like multimerization of that 5 kb.
They are not sure whether there's multimerization. So again, this is a huge problem about whether you get repeats or not. But as far as I know, they just insert it, in an arm of an existing chromosome, and they show that it forms kinetochore at that site. Yeah.